We're on. We're live.
We're live, it's official. You're here, you came all this way. Perfect.
You're going to introduce me?
I was going to ask, would you like an introduction? Do you prefer to introduce yourself? Do you have a...
I don't have an introduction of myself,
okay.
So you're welcome to
Here he is.
gather their attention.
Penguin expert is here.
Find my notes.
Perfect. Give me a few minutes just in case.
Sure.
So you heading back to Washington too?
I'm here for this.
Okay.
And then there's the rest.
You should become a motivational speaker. I like this very much. It's so true.
It's amazing. I mean, you know, it's been so regimented with Otterbein. There's all these wonderful periods of time. But
Sure.
it's, you know, it's like,
It's all there.
yeah.
Wonderful. I'm gonna close the back. Hi everybody, good evening. I'm Elizabeth, one of the reference librarians here at the library, and twice a year I get the honor to look at all the sabbatical research going on at Otterbein and pick two speakers that I think would be of interest to our patrons. So of course penguins, no doubt in my mind, we picked that one straight away, because who doesn't love penguins? So a few housekeeping things, restrooms are right across the hall, water bottle refill and drinking fountain if you need a comfort break. And with us today, we have our penguin expert. I've learned recently retired professor from Otterbein. Hello there.
Hello, I am
Hey, Heidi.
co-chair of the FSDC that funded some of this research. Sorry. And hello.
Welcome.
Sorry.
I'm Elizabeth from the library. Thank you for joining us.
Thank you.
So Dr. Lawrance is here tonight to talk about his work involving penguins. As I said, recently retired professor, was in the biology sequence integrated studies program, human microbiology, and advanced courses in molecular genetics and immunology. So specifically research looked at autoimmune diseases, conservation genetics, that's close. Going to share with us a never before seen presentation, new one he's been working on. So we'll turn it over to you. And how are the lights for you, first of all?
I think they're fine.
Okay, and go up or down over here if you'd like.
Over here might be down a little.
Maybe down a little.
Just for visual effects.
Pardon me. I'll let you guide me. You go down a little.
Excellent.
Yeah.
Better?
Perfect.
Okay.
Very good. Very good. All right. And yes, I'm on. Well, good evening. It's nice to be back in Westerville. Hi, Sarah.
Hi, Sarah.
Nice to see you.
Nice to see you.
Yeah, I'd like to start off by thanking Elizabeth and the others at the Westerville Public Library for hosting the talk, and Heidi, I guess, for the invitation. I don't know. I'm not
Well,
sure
I
who
think it
invited.
started with Alan, but the chairing of the Otterbein Faculty Scholarship and Development Committee that funds this kind of research has switched to Janice Glowski and myself. So I'm the rep.
Yeah, good. And of course, I'd like to acknowledge Otterbein for making this work possible, in particular for providing the students that did the work, and the distinction and honors programs that encourage students to do research as part of their educational experience. The sabbatical leaves program that enables faculty to pursue their research interests from time to time in between all of those classes, those wonderful classes, which, yeah, I miss a bit. And I'd also like to acknowledge Otterbein for, and I'll just say it, for its commitment to the liberal arts and to interdisciplinary learning and education. And in that spirit, in that interdisciplinary spirit, I'm going to begin with a poem. This poem is called Penguin Transitions, and as the name implies, it's a poem about penguins. The author is anonymous, but it's contained in this wonderful book by Rory Wilson, who's a professor of aquatic biology at Swansea University in the UK, and his colleague, David Ainsley. And this is meant to be a little mood visual to help you enjoy penguin transitions. Can you guys hear me all right? Am I speaking now? Silver dimples, restless blue, oceans heaving, darkness through. Monstrous power, but fickle whim. Best sing praises, swells and hymn. Warm life came in times long gone. A bird, a being with flighted song. Merger plea, but ice heart see. You'll need to change to ride with me. Gentle morphing, nuptial vow. Such sweeping change, ethereal now. Unfettered diver, vortex kiss, and sheen sublime. What spirit this? Weaves soothing spells with measured beat, speed spins, cavorts in shadows deep. Twirling, whirling, bubbles make the incantations in its wake. What games are played, what fusions made, in depths beyond the darker glade. In pressured paths for would-be fish, imagined waltz, abyssal wish, but bubbles burst and eddies tire, eroding paths, soon ash from fire. The void has called, snatched ocean's song, the murmur past, the mage now gone. Does anyone here love penguins? Tell me about it.
I've never met one, but I am overtaken by their resilience and their
Yeah.
perseverance.
I've never met one either, but I do think they're super cute.
They are cute. What else?
I've met one and I was able to touch it. It was so cute.
Definitely cute.
I love penguins since I've been a little girl. And I have this huge collection of penguins and I still
Wonderful.
love them.
Yeah.
I was able to kind of hug it and that was my goal. Was hug a penguin. It was at the Long Beach Aquarium.
Long Beach.
Yeah,
Okay.
and
Yeah.
you can go and inside their habitat and hang out with them and feed them.
they are ridiculously charming. And a lot of the qualities they have, perhaps we anthropomorphize with respect to them a little bit. You know, we say they look like they're wearing tuxedos. But they do have a lot of qualities, many qualities that humans share. Resilience, for example. And their mating system, Sarah, am I correct? They have a K strategy. They put lots of energy into a small number of offspring, like we do, rather than whatever the other strategy. Did you know the opposite of that? R, yeah. Yeah. And they're monogamous like we are.
Sometimes.
I'm laughing. Well, they're serially monogamous. They're at least...
Can I take pictures of you for stuff?
Of course. They're monogamous at least for the breeding season. Yeah. And they have a superpower. So I took this picture at the Mystic Aquarium in Connecticut. These are South African penguins. You can sort of make, out the band here. It'll show up better in some other pictures. Yeah, they have a superpower. Penguins most likely evolved from flying seabirds that first evolved the ability to catch fish by diving and subsequently lost the ability to fly as they adapted to the aquatic realm. So their wings are adapted as flippers, their legs are shortened to streamline their shape, improve their swimming. They're also better for sitting on eggs. And their feathers have become more scale-like, more like fish. So they're excellent swimmers and divers. It's as if they can fly in water, upwards of 25 miles per hour, up to 1,000 feet deep, and nearly 50 miles per day. And they originated somewhere around here, Australia, New Zealand. The oldest penguin fossils are about 60 million years of age, and subsequently they diversified all across the southern hemisphere. So as far north as the Galapagos Islands. Did you see penguins when you went to the Galapagos? Yeah, I swam with them. Oh, wonderful. On the equator here. The penguins that I'll be talking about mainly are the South African penguins on the southern coast of South Africa and Namibia here. They don't seem to want to go farther north. No penguins north of the equator, except in the zoos. This is from a paper by Juliana Fiana and colleagues, which worked out penguin phylogeny based on DNA in 2020. And so this is a phylogenetic tree. If you're not used to looking at these, on the horizontal axis here we have divergence, and on the, sorry, did I say horizontal? That's vertical. Vertical axis we have divergence, and on the horizontal axis, time. And so the common ancestor of all penguins is pegged in here back at about 24 million years ago. And the common ancestor of the African penguin.
Please come over to the front desk. For him out to the front desk, please. Thank you.
These are the four species of banded penguins: the Galapagos, Humboldt, African, and Magellanic are in the genus Phoniscus, which appeared just a couple of million years ago. So here you have you can see the banding pattern again of the South African penguin here. Of course, during the time that penguins were evolving, so was our species. This phylogenetic tree, the axes are reversed, so time is on the vertical axis, divergence on the horizontal axis. The last common ancestor of humans and chimpanzees, 5 to 6 million years ago. The last common ancestor of the Denisovans, the Neanderthals, and our own species, what is that, about 500,000 years ago. Just think about that for a minute. All that Neanderthal, Denisovan, sapiens evolution has occurred in quite a short period of time as compared with what we're talking about here. We're going back 24 million years for the common ancestor of all currently existing penguins, and 2 million years for the genus that we're focusing on. This is so fascinating, all of this going on. But coming closer to the present here, so another phylogenetic tree, again, divergence here on the vertical, time on the horizontal. And the origin of modern Homo sapiens, again, somewhere in the range of 500,000 years ago. And then I've got 3 other points marked here. One, the Khoisan people at about 200,000 years ago. The out of Africa people, the people who headed north into Europe and so forth, about 100,000 years ago. And then basically now. So the first encounter between humans and penguins occurred about 200,000 years ago. Right? And the Khoisan people migrated from Central Africa down to the coastal regions. The second came much later when the out of Africa folks who left Africa about 100,000 years ago headed north to Europe, and then from Portugal first headed back down south to the Cape of Good Hope, the southern tip of Africa. And that's what 2b is indicating, because even though that happened 500 years ago, 500 doesn't fit on this scale, right? It's just a blip. We think of 500 years as being a very, very long time, but in fact, that's just in our heads. So we're going to look at these two encounters. I sort of apologize for this picture, but it makes a point. It comes from a 7th grade reader, history primer from South Africa, that is actually entitled Colonization of the Cape in the 17th and 18th centuries. As I mentioned, the Khoisan reached the southern coast of Africa and first encountered penguins about 200,000 years ago. These people hunted penguins and gathered their eggs for centuries. And today the San people continue to have strong relations to the ocean and the coast, somehow managed to live through, until very recently, in a very harmonious manner in which nature is appreciated, respected, honored, and revered. This is Bartholomew Diaz, Portuguese explorer. He's a representation of him. I've actually got this from a travel website, so I couldn't find an actual source for this. I apologize for that, but both of these are kind of memes in a way, and are suggestive of all sorts of things. As I mentioned, the out of Africa people moved north into Europe about 100,000 years ago, leading to a second human encounter with penguins that occurred only 500 years ago. Bartholomew Diaz reached the Cape of Good Hope in 1488, which again, on that time scale, is right there.
Yeah. Just imagine.
The first European settlement in southern Africa was established by the Dutch East India Company in Table Bay, which is part of Cape Town today, in 1652. It was created to supply passing ships with fresh produce, and the colony grew rapidly as farmers settled to grow crops. And as European colonization around the Cape grew, so began the steady decline of penguins, which has occurred over the past 250 years. Mass exploitation occurred when early explorers, sealers, whalers, and fishermen turned to penguin colonies as sources of energy, fresh meat, and eggs. The extraction of oil from penguins' fat layers became economically important in the 1800s and early 1900s. Oil was used for lighting, tanning leather, and fuel. Egg collection occurred from 1875 all the way up to 1968, with the largest annual harvest recorded in 1905 when 62,500 eggs were collected. And as agriculture developed in South Africa, guano, which is the chalky deposits of centuries of seabird excrement, which penguins use to dig in and make their burrows, so as to provide them with shelter from the sun and predators such as gulls, was scraped from the coastal islands for use as fertilizer. From the middle of the 19th century and for about 150 years, in fact, until 1991, 200,000 tons of guano were removed from the South African islands. More recently, oil spills such as the MV Treasure oil spill in 2000 killed thousands of penguins, further degrading their habitat. Recently, the overfishing collapses, there was one in 2001, another in 2013, of populations of mackerel, anchovies, and sardines on which the penguins rely has caused mass penguin starvation. And ongoing climate change has created, they're calling here ecological traps. The fish have migrated to different waters as penguins attempt to fish in their historical fishing waters, finding there are no fish there. And climate change also causing more severe weather events such as floods and heat waves, leading an increased number of penguins to abandon their eggs to seek refuge. And finally, as humans have increasingly encroached on natural areas and spread pathogens around the globe. New infectious diseases are having a devastating effect on the penguins. Avian malaria, avian flu, and avian cholera have all been recorded in the declining populations of penguin species, including those in South Africa. All told, over the past century, African penguin populations have plummeted, dropping from around 1 million breeding pairs in the early 1900s to less than 10,000 in 2023. As you can see, projected out, we could anticipate potential extinction within the next 10 to 25 years. African penguins have been listed as endangered on the International Union for Conservation of Nature's Red List of Threatened Species since 2010. So, the news is not good. In response, however, the Association of Zoos and Aquariums has developed the African Penguin Species Survival Plan. This is one of nearly 300 species survival plans that have been developed by AZA for different endangered and threatened species. The South African penguin species survival plan includes more than 900 penguins, African penguins, throughout 27 AZA accredited zoos and aquariums worldwide. The goal of the SSP, Species Survival Plan, is to maintain a genetically diverse and self-sustaining ex situ South African penguin population. This is done by carefully managing and breeding individuals within the captive population with the goals, obviously, of preserving the species, but also conducting research, educating the public, and potentially reintroducing the species into the wild. Keywords here: in the program's goal are self-sustaining, meaning that they're healthy and reproducing, and genetically diverse. That's where my interest as an immunogeneticist comes in. These are penguin chromosomes. They're actually Magellanic chromosomes. Not seen a picture of South African penguins, but they're very similar. Magellanic have 68 chromosomes. South Africans have 72, and in case you're interested, the sex chromosomes are Z and W, and unlike humans, ZW is female, ZZ is male. Chromosomes come in pairs, just like humans, but no two are exactly alike. Gene variants between any given gene on the chromosome are called alleles. And this allelic variation is key to species survival. It's the raw material of evolution. It enables species to respond as environments change. One particular type of allelic variation is MHC, we call MHC polymorphism. MHC stands for major histocompatibility complex. This is an MHC protein, which is encoded by a gene located somewhere on one of those chromosomes. You're looking at a ribbon diagram which illustrates the alpha carbon backbone of the protein, showing the way the amino acids within the protein are oriented with respect to one another in three-dimensional space. And I think you can see how these helical regions of the protein, together with what we call this a beta sheet region, this flat region down at the bottom, forms a kind of groove. It's almost like a hand, palm on an arm, and it's holding something. That reddish pink purple molecule, which as it goes round, you'll notice it's not actually covalently linked to the MHC protein. It's just being held in the palm of the protein. Immunologists will call this the peptide binding groove, and what's in this groove, or this pocket, is a fragment of a pathogen. The function of the MHC molecule is to present that fragment of the pathogen to the immune system and activate the production of antibodies and T cells. And the remarkable thing about MHC molecules is that they're able to hold on to a wide variety of different pathogens, but not every pathogen. And so what has evolved is a diversity of MHC molecules. Many different allelic forms of MHC. Each individual can only have 2 alleles, right? Because you only have 2 genes, 2 chromosomes. But in the population, many alleles are distributed. So MHCs are very unique in that respect. You know, one of the reasons why we can't transplant organs from one individual to another willy-nilly. You know, a large part of the reason some people get autoimmune diseases and other people don't. A lot of aspects to this diversity that are problematic, but the advantage is that a population that contains a diversity of MHC molecules, even an individual that has at least 2 different ones, is able to respond to a wider array of pathogens than would otherwise be the case. I like to make the analogy of MHC molecules being like canyons. You'll allow me that. This is a canyon which I'm not even going to try to begin to pronounce. Fjadrag Ljufur. It's in southern Iceland. If you are a Justin Bieber fan, apparently this was the backdrop for one of his videos, so you might recognize it. It's about 2 million years old. Why do I make this analogy? Well, like MHC molecules, they have complicated shapes. There's sort of a groove structure here that's containing something important here, a river, just like the MHC is containing a peptide. Like MHC molecules, canyons are also diverse, many different 3-dimensional shapes. These are top-down views of MHC molecules, and they look very similar, but in fact, if you look closely, you can see distinguishing features. So like canyons, MHC molecules are diverse, each with its own unique 3-dimensional shape, holding unique objects within. And as canyons are distributed around the world, so are MHC alleles distributed in populations. But remember, each individual can carry only up to 2 MHC alleles. The total diversity, which may be hundreds or thousands of alleles, is distributed in the population. And together, that distributed diversity ensures that individuals and populations can activate robust immune responses against a wide variety of pathogens. Also, like canyons, MHC alleles are ancient, but actually they're way more ancient in many cases. Some MHC alleles are hundreds of millions of years old, older than the Grand Canyon. We know this because we often find the same allele structure in a penguin species as we do in an amphibian or in a mammal. We call this trans-species evolution. What it reflects is that those alleles evolved hundreds of million years ago, and that's why they're shared in different species. And this, of course, makes them especially valuable from a conservation standpoint. The problem is that species experiencing reduced population size are prone to lose their genetic diversity through a process called genetic drift. So this hourglass shape represents a population before the size is reduced, and you can see there's lots of diversity represented by the different colors and shapes and so forth. But if the population is reduced in size, a lot of that genetic diversity is lost, and even if the population expands again, it doesn't have all the diversity that it had in the past. And it's not likely to regain it because of the fact that these alleles are so ancient, they're so old, they don't evolve back. So, science. We set out to measure MHC diversity in wild penguins and to see how well it is reflected in our SSP population. Our null hypothesis, H sub 0, is that Spheniscus demersus, that's the Latin name for the South African penguin, that they are not significantly different from in the wild population as compared with the SSP population. H1 might be that the wild is more diverse than the SSP, or conversely, that the SSP is more diverse than the wild population. We have nearly 300 penguin samples from the wilds provided by SANCOB, which is the South African Foundation for the Conservation of Coastal Birds. We have 27 from Boulders Bay, 27 from Betty's Beach, 115 from Dyer Island, 97 from Dassin Island, and 29 from Robben Island, which is the island where Nelson Mandela was in prison. And then we have the penguins from the Maryland Zoo and the Mystic Aquarium, which are part of the species survival plan. Total of more than 926 penguins. And from blood samples, we extracted the DNA. We got excited and labeled this one. There it is, the African penguin genome. And we use this technique called gel electrophoresis to analyze the isolated DNA. Gel electrophoresis, some of you may have had an opportunity to experience. It separates DNA molecules on the basis of their size. So small molecules, when they're loaded into these gel wells, will move rapidly through the gel, whereas larger molecules move more slowly. And you can see the effect of that here in these marker lanes, where a variety of different samples of known sizes are separated from one another. And then we have a couple of controls here, but our penguin samples 1 through 5, we have the genome here. In this gel, they all look the same, which is not really what we're looking for. We're looking for genetic diversity. Right? But that's okay. Everybody's MHC is roughly the same size. What we want to analyze is, or the question we want to ask is, do they have the same DNA sequence, right? The same order of A's, G's, C's, and T's within this DNA fragment. We used a couple of techniques to look for that diversity. One of these techniques is called denaturing gradient gel electrophoresis, or DGGE, which separates fragments in a gel on the basis of their melting characteristics, which is dependent on their DNA sequence. And when we put these penguin samples on the DGGE, that's where we're hoping to find diversity, if it's there. So here are 13 South African penguin samples analyzed by denaturing gradient gel electrophoresis, which, as I just mentioned, separates DNA fragments on the basis of their sequence or their melting behavior. And I want to ask you, what do you see here? What do you notice in this image?
I'm going to leave it to my colleagues in the back.
Okay.
They're way more savvy. I see 2 and not 2. That's
2 and not 2.
Yeah.
So, for example, not 2
Yeah.
and 2.
Yeah.
Very good.
And not 1. That's like.
And not 0.
Yeah.
Yeah.
It's fading.
So,
Not going to
there's a
fade
little bit
away,
of
as
variation.
Jerry said.
Right. So, yeah, I asked Dr. Lehman, I asked you this when I presented you this gel some time ago. How might you interpret some penguins having 1 MHC and others having 2?
That.
Those are homozygotes versus heterozygotes.
You're on.
I learned, didn't I?
Of course. Why didn't I think of that?
Right. Some of them are homozygous, some of them are heterozygous. What else do you notice in this picture?
Can you say there are roughly like 4 kind of bands of different positions just scattered throughout there?
Yeah, absolutely. So there's
Wow.
one at this position, there's one here,
Yeah.
there's one here. Gets a little rough to decide about these, but they look like there's, yeah. So in terms of diversity, there is some, yeah?
Good.
They're not all the same. Yeah, so I'm excited. I'm pumped. They've got genetic diversity, right? And they're harboring it in a way that Gregor Mendel would approve of. Yeah, they're either heterozygous or they're homozygous. One of the we mentioned that it's probably better for an individual to be heterozygous, right? To have 2 different MHCs rather than just to have 1. Gives that broader pathogen ability to recognize and respond to. So are there more heterozygotes here or more homozygotes? More heterozygotes, yeah. Yeah. So heterozygote. Well, actually only a couple of homozygotes. So heterozygotes seem to have an advantage. How about, are some alleles more common than others? Yeah, this one at the top is pretty common, whereas this guy, I think he's the only one in this group, right, that has that. So we have some rare alleles here, which also fit in with our notions of how MHC evolves. We have this idea of balancing selection that selects for heterozygosity over homozygosity, and the notion that rare alleles provide an advantage in some situations. So here are members of our species survival plan population, and each one of these penguins has a different MHC genotype, and most of them are heterozygous. So good for them. They're in good shape. As a group, also, not everybody has all of them, right? They're distributed. But, they're as individuals and as a population in good shape.
Simon, did you poke those guys with that thing in their wings, or is that just a visual? Or are they carrying that
These
around?
little guys?
Well, though, yeah.
These are their little marker bands. I mean,
Okay.
you should be able to identify them just by the spots on their chests. But,
Just wondering what I was
make it easier for the management.
as a sociologist.
Yeah.
Oh.
I'm not big on that. I wouldn't want to be walking around with it. So this is the poster that we presented last year, again with the support from Otterbein, at the 11th International Penguin Congress in, oh gosh, I forgot the name of the town, Viña del Mar, just near Valparaiso in Chile, which was a wonderful experience. And major conclusions of our study, just run through these really quickly. We successfully obtained MHC genotypes for 24 SSP and 24 wild penguins. We detected 7 distinct MHC alleles, including some that had not previously been reported. We have data that's consistent with H0, our null hypothesis, but also some concerns that some alleles are not represented in the SSP population, and some that are in the SSP population are present at very low frequency and therefore at risk of loss by way of genetic drift. As you all have noted, heterozygotes predominate, consistent with heterozygote MHC advantage. And that CAPE1 allele is really predominant in both of the populations, suggesting that there is ongoing positive selection. We have actually sequenced the alleles, that's another part of this study, and found that they're shared with other banded penguins, consistent with this notion of trans-species evolution, and again emphasizing the significance of conserving these special molecules. Major recommendations of our study: the data is being used to set up the breeding of the penguins to maintain this genetic diversity, avoiding genetic drift. We'd like to expand the studies to include other SSPs, do some more work to understand the immunology a little better, do it more efficiently and with less invasive techniques, figure out how we can do this for diversity of populations that can't be managed through SSPs. We've done some work with mountain gorillas and looking to do some work with orcas in the future. Integrate with other studies of genetic diversity, and of course work to end the overfishing, oiling, climate change, and emerging diseases that are causing the problem in the first place, which is a tall order, but hang on folks, we have ended the harvesting for meat and oil. We've ended the egg and guano collection. Things can get better. We have to push on the arc.
On the arc.
Push on the arc. The arc of justice.
That's right.
Yeah.
All day long.
So for the moment, these guys are okay. Not in the best of situations, but not in the worst. So I'd like to acknowledge folks who helped with this project, in particular Mike Cranfield, who sadly passed away last year, who was by connection through the Maryland Zoo with Sam Cobb and other wonderful people. Some of Dr. Lehman, Dr. Lisinski might remember Kelly Huth and Courtney Katz, who actually started this project in, okay, this is a month's day, because it was 2010, something like that, when they were with us. Antonietta, who went with me down to Chile to present the work, and this is Tracy Romano and Gail Sapensky from the Mystic Aquarium. And so again, thanks to this Otterbein University Sabbatical Leaves Fund, Student Research Fund, other students who've worked on the project, Donna, who's here this evening, and Aaron, and even the students from my penultimate class, I guess, immunogenetics from spring of last year. I'd like to acknowledge with gratitude and honor all of the penguins and the lands and waters that they inhabit, and remember Tracy Spoon, who actually suggested this project years and years ago. Thank you very much. A few science references from the science journal literature, and then a couple of books that I have really enjoyed reading. This is the book that had a poem in it by Rory Wilson and Dave Ainley. Dee Boersma, who's at Seattle, University of Washington in Seattle, and Rachel Carson, who just has a new book out. Maybe suggest that one for the library.
There you go.
It's got kids' illustrations of it's called The Air Above Us, I think. It's about the sky above us. That's what it's called. For me, I have moved to Bellingham, Washington. Where are you, Bellingham? There, right there. 30 minutes from the Canadian border.
Wow.
And the Salish Sea here, which is used to be called just Puget Sound, but now it's got this name that in part recognizes the indigenous cultures that have lived in this area for many, many years. Yeah, this is up Mount Baker, and I've been, since I retired, taking a little time to do a little downhill skiing. But there's a lot of wildlife concerns up there, in particular the orcas. There's a population of orcas that's known as the southern resident killer whale population, and there are only like 70 of them, and they live just in the Salish Sea there, and they're starving to death basically because the salmon have failed. So there's lots of work to be done. So yeah, let's do it. Thank you very much.
Namaste.
Thank you. If I may just interject, we do have the Rachel Carson book.
Excellent.
If anyone would like to, it's checked out with a hefty waiting list. If you'd like to place a hold, give us a call or stop upstairs. Thank you so very much. This was
My
fascinating.
pleasure. Yeah.
Do you have questions or comments?
Dr. Lisinski.
I do have a question, but I'm not sure that, so I'm going to ask it anyway.
Go for
I'll try
it.
to ask it in a nice way. So I thought MHC was this really, really complex thing that we as individuals were different in, and that we could like find our mates because they had slightly different MHCs and stuff like that. So that I thought there was like bazillions of combinations of it. And then here you're saying that each individual penguin only has one allele, right? So
One or two,
which is
right?
it?
Or
Or two if they've got two places, but they only have one per string. So I'm missing, do we have one MHC, or do we have, like, is everybody in the room all very different and we can tell, like, our mate choice is somebody who's got a different MHC, or is it really just
Well,
a few of
mate
them?
choice is another wonderful aspect of MHC that, you know, could be the subject of a whole nother talk. You know, it's, turns out that in many species, individuals choose mates with different MHCs, which makes sense in terms of heterozygote advantage, right? If you choose a mate that has the same MHC, you're much more likely to produce homozygous offspring. So nothing that you've said is inconsistent in my mind with what I presented. So, I mean, yes, we are all different.
Okay, so.
And they are too.
All right. I guess I thought it was more variable than it really is. Anyway.
Well, it is. It's tremendously varied. And remember, I mean, maybe this will help. We're looking at just one small part of the genome here, right? But things are changing. You know, we're getting to the point where, with this nanopore sequencing, we can sequence the whole MHC or the whole genome and get a better picture of the diversity. The DGGE is a pretty archaic technology to the point where Bio-Rad does not support it anymore. But it's a technology that students were able to work with and actually, you know, get data that is significant and makes the point. And so, yeah, that's where we are, or where we were. Where we're going, I'll get back to you on that. Yeah.
So will these penguins breed with who you ask them to breed with?
They can be encouraged. Yeah.
How do you encourage?
Go on YouTube. Ask YouTube. You'll see.
Because there's some that are really valuable.
Yeah.
Right? Some of those birds
Yeah.
are really valuable.
Yeah.
I mean, they're all
Yes.
valuable, but some
And with the
more so than
SSP,
others.
they can move, you know, they move them around from one zoo to another, aquarium to another.
Do they do it artificially? Will they artificially inseminate?
That's possible. Yeah.
Any other questions? Thank you so very much.
My
That was
pleasure.
Simon,
Heidi.
you're hard.
What? Pardon?
Are you here for a while? How soon?
Just for a week.
Well, let's get together.
Okay.
Maybe we can get